Hello everyone, my name is Obiane Fokosi Soshukwu, and today I'll be talking about CRISPR-Cas9, a groundbreaking technology that has changed the way scientists study and edit genes. Before we begin, what does CRISPR stand for? CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. CRISPR allows researchers to make precise changes to DNA, opening the door to new treatments for genetic diseases and many other scientific advancements. In this presentation, I'll explain what CRISPR is, how it works, its benefits and ethical concerns, and share my thoughts on how it should be used responsibly. What is CRISPR? So what exactly is CRISPR? As I mentioned earlier, CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. While the name sounds complicated, the idea is much easier. CRISPR is a gene editing tool that allows scientists to find and change specific sections of DNA. It was first discovered as part of a natural defense system that bacteria use to protect themselves from viruses. Today, scientists have adapted this system to edit genes in plants, animals, and even human cells. Think of CRISPR as like the find and replace feature in Microsoft Word. It finds a specific DNA sequence and allows scientists to repair, remove, or replace it. How does CRISPR work? The process starts with a guide RNA, which acts like a GPS by locating the exact DNA sequence scientists want to edit. It then brings the Cas9 enzyme to that location. Cas9 acts like a molecular scissors by cutting the DNA. After the cut is made, the cell repairs the DNA, allowing scientists to replace, remove, and correct a gene. This precise process is what makes CRISPR such a powerful gene editing tool. And as you can see on the diagram, how I connected how CRISPR works. The molecular scissors. Cas9 is one of the most important parts of the CRISPR system because it performs the actual cutting of the DNA. In Biology 101, we learned that enzymes speed up chemical reactions without being used up. Cas9 is a perfect example because it cuts DNA at the exact location identified by the guide RNA. CRISPR in action. This image comes from the assigned CRISPR video at about a minute and 41 seconds. At this point, the guide RNA pairs with the target DNA sequence, allowing Cas9 to locate the correct spot before making the cut. This animation helped me better understand how precise CRISPR can be and also how accurate it can also be. Benefits of CRISPR technology. CRISPR has many exciting applications. Researchers are studying it to treat genetic diseases, develop new medicines, improve crops, and better understand how genes work. One of its greatest potential benefits is correcting harmful genetic mutations that occurs in inherited diseases. And as you can see on my diagram, I've showed how the benefits of CRISPR. Ethical concerns of CRISPR. Although CRISPR offers many benefits, it also raises ethical concerns. Off-target edits could accidentally change the wrong part of DNA, and some people worry about using gene editing for non-medical enhancements, sometimes called designer babies. Because of these concerns, CRISPR should be used responsibly and ethically. My opinion. I believe CRISPR is one of the most promising scientific discoveries of our time because it has the potential to improve and even save lives. However, it should only be used under strict ethical guidelines. Before I finish, I would like to ask one question. If CRISPR could safely cure a serious genetic disease before a child was born, would you support it? Why or why not? And for my last slide, these are my references. Thank you for listening to my presentation. The information in this presentation came from my course materials, the assigned CRISPR video, OpenText Biology 2 Echo, and the National Humane Genomic Research Institute. I also use ChatGPT to help organize my ideas and improve the wording while reviewing the content for accuracy. Thank you.